Hello, good morning, everyone. Welcome back for another week of biology. So today we will be doing lecture 4, which is membrane transport. All right, so let's get started. Before we fill in the study guide, maybe we could review a little bit about the structure of the cell membrane. So if you want to draw along, please do so. Okay, so first of all, the cell membrane is what we call a fluid mosaic model. Mosaic model. And what that means is, number one, it is not a static structure. Okay, it is something that can continuously be remodeled. Okay, it's constantly changing. That's what fluid means. A mosaic means it has many different components to it. So it's not just made up of one particular thing, but many different things together. And it forms this big sheet that covers the entire cell. Okay, so it's called a fluid mosaic model. So let's draw this fluid mosaic model and see what are some of the things that we are going to be able to find on the cell membrane. Okay, so this is again, this is the cell. Okay, and this is the nucleus, and we are basically zooming on into this part right here, the membrane that surrounds the cell. Okay, so remember, cells, there are three things that are shared amongst all the cell types, right? They all have they all have a cytoplasm, they all have genetic material, and they all have a cell membrane. Okay, and so we are looking at the plasma membrane over here. Give me 1 second. People seem to. Okay, so over here, we would see the major component of cell membrane are these lipid molecules, right? Anybody want to let me know what they're called? What am I drawing here? Oh, these little circles with the two tails. Anybody? Yes, so these are phospholipids. Okay, so phospholipids. Okay, so remember, phospholipids has three parts to it, right? There is a phosphate head. There's a glycerol, and then plus two fatty acids, right? So those fatty acids could be saturated, could be unsaturated. It's going to be a mixture of both, and that is the major component of our cell membrane. Okay, so it forms what we call a phospholipid bilayer. Phospholipid bilayer. So not just one layer, but two layers of phospholipid together. Okay. Now, every now and then, you are going to have some proteins that are inserted in this phospholipid. Okay. So there is going to be a protein, and the protein might go all the way through your phospholipid from the outside to the inside. So if the membrane goes all the way through, we call this an integral protein. Integral protein. Okay. I remember when I was learning it for the first time, I got really confused about this diagram. Like I thought this was the outside and this was the inside of the cell, but actually this is the outside. This is the outside, what we call the extracellular fluid. Okay, extracellular fluid, and then this is the inside of the cell. Inside of the cell. Okay, and this actually two layers, the bilayer, forms the cell membrane. Okay, so again, we have integral proteins that goes from the outside all the way to the inside, and there are many different functions of these proteins. If you watch the lecture recording, you might remember one of the functions of the integral protein is there might be a little channel on the inside, and that would allows things to move through it. So it can either go from outside to inside or the other way around. So there you have it, an integral protein. Okay. What's another component of the cell membrane? Anybody remember from the lecture? Feel free to just shout it out. Tyler said cholesterol, and that is of course correct. So you might have some cholesterol here. Remember, cholesterol is a steroid. So there is going to be the 4 fused ring structures over here. Okay. Now, but by the way, I should mention this lecture, as well as lecture 1, the scientific method, these are very related to the lab that you have to do next week. Okay, so pay extra attention to the stuff I'm saying, and maybe you want to rewatch this afterwards because you might find some answers to some of the questions in the lab. Okay, so these are again cholesterol molecules, and the function of the cholesterol. I think we have a question coming up for this thing. Nope. Okay, so I'm gonna write here. Okay, cholesterol. The function of cholesterol is to adjust the fluidity of the membrane. Okay. Remember, this is not a solid wall. If it's a solid wall, then nothing goes through, right? And the cell's going to die basically because you cannot exchange material with the surrounding. So it's more like think of it kind of like a soap bubble. Okay, if you go gentle enough, then you can go through the soap bubble, right? So how fluid is it going to be? Okay, that depends on how much cholesterol you have. So more cholesterol equals to more fluid. Okay, so very, very leaky then. Okay, we don't want to have too much cholesterol because then everything would just leak outside, everything would just go inside. There's no control, right? No regulation. If you have less cholesterol, okay, then it's going to be more solid. We don't want it to be too solid either, right? Because then again, like I said, nothing would go through. So that's why having the right amount of cholesterol will help adjust the fluidity. Okay, so it depends on the situation, right? Depending on what kind of cell you're talking about. Some cells might want to have more cholesterol, and some cells might want to have less cholesterol. So let's think about this. Let's say there is an organism living in very cold environment. Okay. We can say a plant. Okay, a plant, make it easier to visualize. A plant grows in very cold environment. Okay, northern Canada, Yukon or something where it's like super cold. Do you expect the cell? Okay, do you expect the cell membrane to have more or less cholesterol? Okay, so compared to like a regular cell, okay, that is found in like regular environment. So more, less, or no change. Okay, let's throw in none of the above, as in like not quite sure what the answer is. I'm gonna fold start the poll, and you can tell me what you think. Again, this is the question. I know sometimes I ramble a little bit, but this is the question. Okay, you have a plant that is growing in very cold environment. Do you expect that the cell membrane to have more cholesterol in its content, less cholesterol, or doesn't really matter? It doesn't matter if it's cold environment, hot environment. All the plant cells should have similar cholesterol content. Okay, so about 1/3 of the class replied. Just a couple more seconds in case anyone else want to chime in. Okay, so we are, kind of half between more and less, and then some people think there's no change. Does anybody want to justify their answer? Anybody want to explain to the class why they choose what they chose? Okay. You can either explain it by unmuting, or you can type it in the chat if you want. Anybody want to explain why? I just want to know the logic, right? To see.
I can try to answer this, but I
Yeah,
don't know
sure.
if I'm going to embarrass myself.
No, not at all. Thank you for taking the courage to do that. Go ahead, please.
I honestly think that if a plant grows in a cold environment, that it would have less cholesterol, because the cold could possibly slow the production, and it could slow the fluidity of the membrane. I don't really know, but that's what I thought.
Okay.
So,
yeah, great. Thank you for sharing, Sarah. Anyone else from the other side? So I assume Sarah chose less, but anybody chose more and would like to share their side of their point of views? No. It's fun. Just a quick side story. I watch my kids do online learning, last couple of weeks, and whenever a teacher asks a question, everybody, click the raise hand button and let it pick me, and then my daughter would get upset when she doesn't get picked. And then when you come to do teaching at the college, when you ask a question, it's like dead silence, right? Nobody wants to say say anything. It's completely opposite. Anyway, that's okay. Let me explain what the correct answer is, so that you know what to choose on the test, or in the lab or whatever, right? So if it grows in a cold environment, okay, forget about cholesterol for a second. If it's in a cold environment, do you expect the cell membrane to be more solid or more fluid due to the cold? Well, if you think about it, right, a molecule slows down as the temperature drops, right? So in a cold environment, naturally the cell membrane would become more solid. Okay, and again, I keep on emphasizing that you want it to be just the right consistency, not too liquid, not too solid. So to counterbalance the cold environment, okay, to prevent the cell membrane from being too solid, okay, a countermeasure to that would be to have more cholesterol, because more cholesterol is going to make it more fluid, and therefore counteracting the effects of the cold on the cell membrane. I hope that makes sense. Any follow-up questions? Aiden said it's there less cholesterol, it's more solid, it benefits from. Okay, so yeah, it's basically what I said here, right? Okay, so if it's cold environment, you want to counteract that effect of the membrane being too solid, okay, and therefore you would put more cholesterol in the cell membrane. All right, so here is the takeaway point. No two cell membranes are going to be identical. All right, if it's the same type of species, of course they're going to be similar. Okay, but if you're talking about like two different organisms, right? Perhaps they grow in different environments. They are going to have different composition. All right, so don't expect the cell membrane of, say, a cactus, right? It's going to be identical to that of watercress or something like that. All right. So of course the converse is true. If the plant grows in a very hot environment, right, like a desert plant, then you would expect it to have less cholesterol because it's so hot. The membrane is already so fluid, right? You want to counteract that by having less cholesterol. I hope that makes sense. Okay, moving on. In addition to integral protein, there are also some of these proteins that are just kind of, sticking on either on the outside or sticking on the inside. It doesn't span the entire cell membrane. It's just either on the outside or in the inside. These are called peripheral proteins. erythroproteins. And again, they have various functions. And one of the functions is it enables the cell membrane to kind of attach to these cytoskeleton. Remember, we learned about cytoskeleton last time. There are these actin filament, and then there are these microtubules. And by having some of these protein, it provides an anchorage point to attach things. Okay, and just a couple of other small things that I want to mention. The interior of the cell membrane is going to be hydrophobic, right? Okay, because it consists of primarily the fatty acids, whereas the head, the outside portion, and the inside portion, these are hydrophilic. Okay, so anything that's really small, anything that's polar would be able to cross the membrane quite easily. Okay, and just lastly, every now and then, some of the proteins, they might have some sugar attached to it. Okay, if it has sugar attached to the protein, we call it like a glycoprotein. Right, all these is in the PowerPoint, of course. And sometimes you might have lipids that have some sugar on it, right? Okay, and these would be called like glycolipid. Okay, so sugary lipids and sugary proteins. Okay, I know this is kind of like a messy diagram, but if any of you like to have a screenshot of it, take a picture, go ahead before I go back to the study guide. Okay, and of course, if you have a question, please ask. Alrighty, let's go to the study guide now. First question, briefly describe the functions of membrane proteins by completing the chart below. Right, so here is the first function. Okay, let me try to fit all these things in the box. Okay, the first one is the ability. It's basically what I just described, right? It allows attachment to cytoskeleton. Okay, that's the function, right? So allows cell to attach to extracellular matrix. Okay. Now, what does that even mean, right? If you think about a cell here, right, the outside also has a lot of cytoskeleton. Okay, now think about like how a cell can move from one place to another, right? So they need to form attachment to these extracellular fibers, also known as the extracellular matrix, and it basically crawls around. Think of it as like I don't know what the proper name of those things are, but at the playground there are those ropes that forms these like nets, right? And you can climb on it, right? So that's basically like this extracellular matrix, and then the person would be like a cell just moving along on this grid, right? So every time you grab onto a rope and pull yourself, the cells don't have a hand, but instead they have these proteins that allows them to grab onto the extracellular matrix, and then they move around. Okay, so that's one of the functions. Now, another function is not only do you attach to the outside, you also attach to the inside, right? So that would allow the cell to maintain and support cell shape, right? By providing anchorage point for cytoskeleton, for actin, for example, right? And intermediate filaments, right? Okay. So that's the first function of proteins. The next one is cell signaling. Cell signaling. Okay. So they act as receptors. Receptor for proteins. Okay. Once the correct protein binds, it will trigger a series of events that will change the cell's behavior. Okay. So an example of this would be protein hormones. Protein hormones. All right. Here is our lovely cell. Okay, and then this is the nucleus. And then on the surface, we are going to have a membrane protein that acts as a receptor. Okay, so each receptor is only going to allow certain types of molecules to bind. Okay, so if we're talking protein hormones, it will only allow proteins of a certain shape to bind. And as you can see here, this receptor will only allow the triangle protein hormones to bind. So once this binds to it, okay, maybe I use a different color. Okay, so this is going to come here and bind to it. As soon as this occurs, you're going to have a series of reactions going on in the cell. And ultimately, this is going to perhaps turn on a gene or turn off a gene. Okay, so then it changes the behavior of the cell. So in the lecture, these series of events, these series of reactions, it is called a signal transduction. Signal transduction. Okay. Now this is for protein hormones. Steroid hormones are different. Okay, steroid hormones can go directly into the nucleus. You might wonder why. How come hormones can go through? Remember the cell membrane, I said earlier, it is mostly hydrophobic, right, in the center. So proteins, their surface are typically not hydrophobic, right? All the hydrophobic stuff are in the core, and they're very big, so they cannot really go through. Steroid hormones, on the other hand, steroid, as you know, it's a lipid, so it's hydrophobic. So if something is hydrophobic, it's able to cross the cell membrane very, very easily. Okay, so there you have it. That is the second function. Any question? Okay, so here we can see, right, that is the molecule that is going to bind. We actually have a name for it. I'm just going to pull it down. You don't have to know it, but you might hear me say it, like, you know, next semester or something. This is a ligand, okay, or ligand, depends how you want to pronounce it. So a ligand is basically the correct triggering molecule for a particular receptor. Okay. All right, another function. Let's go back to purple. I like to switch back and forth. This is enzymatic activity. Enzymatic activity. Okay, you might remember an enzyme is protein that allows you to speed up chemical reactions in the body. Okay, so sometimes we want to carry out multiple reactions close to the surface of the cell membrane. Okay, we do that for signaling purposes. We do that for immune purposes. Okay, and then it's just convenient to have multiple enzymes that belong to the same pathway to be so close to each other. So what do I mean by that? You know, let's say I have A, okay, some kind of starting material A, and I need to convert it to B, and then I need to convert it to C. In order to do this, I am going to need enzyme number 1 and enzyme number 2 for the second reaction. So rather than having these enzymes like randomly flowing around in the cytoplasm, why not just have them one next to each other on the cell membrane? So, you know, A comes, right, and number 1 is going to convert into B, and immediately the B can be picked up by 2, and then it converts it into C. Okay, so this allows chemical reactions to occur rapidly. Okay, you can say multiple chemical reactions, series of chemical reactions. Okay, so because it's one after the other, right? Usually these enzymes, belong to the same chemical pathway. Okay, it's like the same assembly line. All right, you want them to be close to each other as much as possible. Next function. Okay, this is called intercellular junction. Right? Remember we talked about this last class, I think, when we discussed intermediate filament. Right? Intermediate filaments are like staples that cause two cells to be stuck together, right? So I mentioned, you know, in the intestines, you want the cells to form a tight seal, so nothing goes through them, right? So in addition to the intermediate filaments, we also need to have some of these proteins, membrane proteins, to help us, you know, seal the connection. All right. So membrane proteins of adjacent cells can hook on to each other to form a junction, a connection, a sealed, so nothing can pass through. Okay, so these junctions prevent uncontrolled movements of substances between cells. Okay. You will find these again, like intestinal cells and the kidneys. When you filter your blood to produce the urine, you don't want the material to leak back into the blood, right? And then you only want certain materials to go from the blood to the urine. And so you need to form these tight seals between the cells. So anything that does get through is done on purpose rather than, you know, because of leakage. So that is the cell junction. Okay, and in case if you are a little bit confused about this diagram, this is cell number 1, and this is cell number 2. 2 cells, and then these are the membrane proteins, and they, you know, lock onto each other. Next one is what we call cell-cell recognition. How does the cell know what belongs to the body, what does not, right? So this cell-cell recognition is a key feature of the immune system. You're going to learn about the immune system next semester, and your white blood cells, for example, would have to check what is on the surface of each of the cells. And if it sees something that doesn't belong to the body, then they recognize it and it will attack it. Okay, so in this case, this would be a white blood cell, and then this would be like another cell. Okay, so you see we have the protein here, and then we have some sugar, right? So earlier I told you what this is called, right? It's called a glycoprotein, right? A sugary protein, right? So there are many molecules on the surface of a cell. Okay, so these molecules, all these molecules on the surface of the cell, we call it antigen. Antigens. Okay, so you have antigens, I have antigens on my cell. Bacteria, they have antigens. Viruses, they have antigens. Okay, and so the white cell's job is to check to see if those antigens are the same as the ones in your body. Okay, if it's not, then it knows it's foreign, and then it will attack it. Okay, so many of these antigens are protein based. Okay, they're glycoproteins. Sometimes it could be glycolipids as well. Okay, so membrane protein, one of the function is enable cell-cell recognition. Okay, just gonna write one more point here. WBC, white blood cells, will attack anything with foreign, antigens. Okay, foreign means outside of the body. Okay? I want to emphasize antigens are not necessarily bad. Okay, antigens are just these molecules on the cell. Right. So again, you have your antigens, I have my antigen. Okay. But then if I take my kidney and then donate it to some random person, okay, that's not necessarily going to work, because on my kidney, the cells will have different antigens than the person who's receiving it. Okay, so the person's immune system will attack and reject the kidney. Okay? That's why for transplant, you have to make sure that there is like matching antigens, right? It's not possible to have 100% match. Okay, the only time where you have 100% match between two individuals would be what? Anybody know what I'm talking about? When would you have exactly the same antigen as another person? There's only one unique situation that will allow that to happen. That's right, identical twins. Okay, very good. And so if it's not twins, then you would not have the same antigens. You might share some antigens between you and your siblings. Okay, that's why people who are related usually would be considered first in organ donation. Okay, but like I said, you won't have 100%. You will have anywhere between 50 to 100% between you and your sibling. Okay, but only if you have an identical twin would you have 100%. Okay, just random thing. I like to talk about random stuff in the lecture. I read last week that they did a first successful heart transplant to a patient using genetically modified heart from a pig. Okay, so they're able to genetically alter the surface antigens of the cells to minimize the rejection. Okay, so this is called xenotransplantation, right? From animals to humans, and if this works, then it's going to open up a completely new door to organ transplant. As you know, organ transplants are very important, and there are many people on waiting lists. Anybody know what's the most the organ that is in the highest demand for organ transplant? Anybody want to guess what that might be?
Is it like the lungs?
Lungs is it's one of the top
Yeah, 'cause I
5
know
for
it's a lot
sure.
of people like smoke and stuff, and then it can cause them to need something like that. But
Right. Yeah. So lung is definitely one of the top ones, but not the most common one. A lot of people say kidney in the chat. Yeah, kidney is the most common one. The wait list for kidney is very, very long. And I actually teach another course that has a topic on organ transplant. And I wanted to see how difficult it is to become an organ donor. In fact, it's actually very easy in Ontario. Anyways, you go to the website. You can just type in Google like Ontario become a organ donor, and it will take you to the Trillium of Life website, and then you just register. Okay, you need to be 16 and older. You need to have a health card. And you need to give them your consent. And I was able to become a donor, literally within 5 minutes. So if that's something that you're interested in, definitely check it out. My philosophy is when I died, might as well, let my body help anyone who could benefit from it. But my wife always says, nobody wants your organs. Nobody wants your eyes. You have terrible eyesight. And I tell her, take whatever they can. They don't have to take they don't have to take my eyes. They could take something else. Anyways, that's something that you can think about. So last one is transport. Okay, transport, and that's kind of like the focus of this lecture, right? So transport is the membrane would form channels, okay, and carriers to allow materials to move through the cell membrane. Okay, so things that otherwise would not be able to cross through, then we would rely on these transport proteins. So, for example, large molecules, right? Charged molecules. Okay, and polar molecules. They would need help using transport protein. So there you have it. Those are the functions of the cell membrane protein. Anybody have any questions about any of these before we move on? All right, so let's see if we have any poll questions. Oh, not yet. These are for later on. So let's go on. Oh, by the way, since most of you are here already, let me quickly talk about next week before I lose any of you. Next week there will be no tutorial. Okay, next week there's no tutorial. There's no new lecture. Okay, you don't have to watch anything. You just have to do the quiz. Okay, so here's the quiz number 2 information. I'll post this online afterwards, of course. It's going to happen on Tuesday, and then you have the usual window from 6 to midnight, 45 minutes. And this time is on lecture 3 and 4. Okay, and 22 multiple choice question, 2 multi-select questions, and 4 application questions this time. Okay, application questions is worth only 1 mark each. They are not like super hard. We'll do some examples later on. Okay, it's going to be 10% and out of 30. Right. I got some emails from students asking how come some they're not sure why some questions have more than 1 answers. Okay, so those are the multi-select question, right? And you can always tell it's a multi-select question because number 1, it's going to be out of 2 marks, and number 2, the question will say select all correct answers. Okay, and it doesn't really matter because if it's a regular question, you won't be able to select more than 1 answer anyways. Okay, so in case there's any kind of confusion, that's what it is. All right, and then the second thing is next week, again, no tutorial, right? Instead, you'll be working on the lab. The lab is going to be available. Actually, I think I'll make it available today. Okay, and then towards the end, I'm going to show you how to download the lab and then how to submit it. And I'll give you some pointers for the lab as well. The lab is like 10%. Should be an easy 10% for you to get if you follow the instruction carefully. Okay. So any question about the quiz or what's going on next week? Yeah, great. Do you guys know you can do like a thumbs up on the in the chat box or something? Not chat box, but like in the on your Zoom thing. So I know you know. So sometimes Maybe you can give me some of those like feedbacks. Yep. Okay. I see some thumbs up. Very good. Okay. Next, we have this thing over here. Complete the following diagram to illustrate. Allison gave me a turtle. Okay. I don't know what that means. Complete the following diagram to illustrate the relationship between anabolism and catabolism. So just to help you, define these words a little bit because it can be confusing. Metabolism is basically all the reactions in the body. Okay, so we are basically like a flask with multiple reactions going on in the body. These reactions help us, stay alive, right? We have reactions to digest our food. We have reactions to, boost up our immune system and so on and so forth. So all these reactions collectively together is called metabolism. Okay, now there are two types of metabolism. There is anabolism. Okay, anabolism in its simplest terms is basically building things. Okay, so reaction that involves in putting things together. Okay, building molecules. We call that anabolism. The other one is catabolism. Catabolism, the root word, right, is derived from like the same meaning as catastrophe. So in catastrophe, everything breaks down, right? So catabolism is breaking things. Breaking things. Okay. So these are the two types of metabolism. Right? So over here, in this diagram, these are like macromolecules from our diet. We ate them. Okay? So macromolecules from food. Okay? So you ate a sandwich. Okay? You're gonna have some carbohydrates. Maybe you slap on a piece of cheese, then you're gonna have some lipids and some proteins. All these things are macromolecules. We are going to break it down in our digestive system. And then ultimately, it's going to be picked up by our cells. Okay? So we will take these macromolecules, these polymers, if you would, okay? And we will break them down into many building blocks for biosynthesis. Okay, so these are basically monomers. Okay, so we're going to use these for making biological molecules. That's what biosynthesis means. So this process of breaking polymers back into the individual building blocks, right? Again, we are breaking things. So that is called the catabolism. Okay, catabolism or catabolic reaction. Okay, and to break things, it is actually going to release heat. Okay, we are actually not very efficient as an organism. A lot of the energy we use to break things down, to build things, actually is wasted as heat. Okay, so it generates quite amount of heat. It's not completely wasted, I guess, because we do need to maintain a body temperature of 37 degrees. So after you eat the food, right, you feel warmer, right? That's because you're breaking the food down, the heat. Okay, and other energy that are released will be captured by these molecules called ATP. Okay, so ATP is the usable form of energy in the body. Okay, so let me write this down for you. Okay, energy released from catabolic reactions are captured in the form of ATP. And then when you want to build molecules, you're going to use the monomers that you've got previously. And then you're gonna use the ATP that was capturing the energy from catabolic reactions to build things in anabolic reaction. Okay, and ultimately you're going to end up having macromolecules needed in the body. Okay, so just as an example, right? We need to make insulin, right? Insulin. Insulin is a protein hormone that helps us regulate blood sugar. It's a very important hormone. But how are we going to get the amino acid to build this insulin? Okay, so we're going to eat the food, right? That cheese that you ate in your sandwich, that's going to have amino acids in it. Okay, but it's not necessarily in the right order, right? As insulin. So you break it down. Now you get the raw material back. You get the proteins in the form of amino acid. And then you will take those amino acids and then you build it into insulin. Okay, remember there are only 20 amino acids, right? So you can rearrange them and make it into something that you need, something that is useful. Okay, so that's the relationship between catabolic reaction and anabolic reaction. Okay, in one sentence, essentially, the energy that is released from catabolic reactions is what is required for the anabolic reaction. Okay, so the energy released from catabolic reaction drives the anabolic reaction. It provides the energy to build things. Right? That's why we need to keep on eating, right? If we don't eat, we don't get ATP. We can't do any of these, our cell would break down. Any questions about metabolism? Okay, so what else can we use ATP for? Right, so 3 different kinds of cellular work that requires ATP. Okay, so the first one is mechanical work. Mechanical work. Okay, so these involves like physically moving something. Okay, physical movement. All right, so like muscle. If you imagine running at full speed, okay, as fast as you can, you're not going to be able to do that forever. Okay, if you're good, you do it for like a minute or 2 minutes at full speed. Eventually, your legs going to start to feel sore, and then you're not going to be able to do it anymore, right? So what that means is when you reach the point where you have to give up, that's when you don't have enough ATP for your muscle to move. Okay, and then you have to stop. Right? You have to wait for the ATP stock to be replenished, okay, before you can do that again. So that is an example of mechanical work. Okay, on a cellular level, if you want to say move a cilia or move the flagellum of a cell, that is also an example of mechanical work. Can I have another example of another type of cellular work that requires ATP besides mechanical work? Yeah, Adriana said chemical, and Tina said transport. Very good. So chemical work, right? So such as anabolism, right? That's chemical work, right? Enzymatic activity, right? That requires chemical work. And the last one is transport. Transport work. Okay, so active transport. Moving things uphill, right? Moving things from area of low concentration to high concentration. That is going to require ATP. Okay, let's do a quick poll just to see if you are paying attention. Go ahead. Okay, only half of you have submitted a response. I'm going to give you a couple more seconds. Oh, sorry, my bad. This says select all correct answers. I should have give you the multi-select poll instead. It's okay. Okay. We'll just talk about it. Right, so catabolism, select all correct answer. Consume more energy than they produce. That is not true. Okay. If you look at the diagram we had, like here, right, you are actually producing energy in the form of ATP for catabolic reactions, right? Okay. Breaks down polymers into monomers. That is correct. Produce excess heat to warm the body. That is also correct. Only occurs in human cells and not other cells. That's not true. Okay. All cells have catabolism and anabolism. All right. So those are the two correct answers. Okay. And again, I should have given you the multi-select poll instead. Okay. Let's go back to here. Let's keep going. List 4 factors that affects the rate of diffusion. Okay. Anybody can help me out here? Yeah, pH, yeah, certainly it could in some instances, although not one that we talked about in class though. Temperature, yes. Okay, so generally, higher temperature, increased temperature equals to faster diffusion. Someone says charge, that's correct. Okay. So let me give you an example of charge. So let's say I have a membrane here. Okay. If I just have molecules like this, okay, then diffusion will occur, right? It will go from top to bottom, right? Okay. But if I have charged molecule like this, like H+, this will happen much faster, the diffusion, because the like charges, they repel each other, right? So like charges repel each other. Okay, so it's going to push it across the membrane faster. However, this will only be true initially, okay? Because as soon as you have more positive crossing over to the bottom, now you have a buildup of positive charge at the bottom, and that will actually create like a counter force that prevents more H+ from going through, okay? Because they want to get away from each other as much as possible, right? The positive charge. So as soon as you have a buildup of positive charge here, then they won't be able to cross as easily, okay? So charges will affect the rate of diffusion. Another one, anybody? Let's see what else people say. Someone say like size, yeah. Okay, so generally bigger equals to slower, right? And steepness. Okay, so what does steepness mean? Let's say you have two sides in the chamber, and this is a semi-permeable membrane. Okay, let's say you have a 70% sugar solution and a 20% solution versus like a 70% to a 60% solution. Which one will have faster diffusion? Okay, the sugar will go from the higher concentration to the lower concentration. Okay, but if the difference is greater, okay, between 70% and 20%, this will have faster diffusion. Okay. Over here, it will be slower because the difference is less, right? Okay, so this difference is what we call a concentration gradient. Okay, the greater the difference, the steeper the concentration gradient, and then the faster the diffusion will take place. Okay. Now, I just want to talk about a question here that a lot of people get it wrong in the past on the quiz, one of the application questions. So I'm going to talk about this. Osmosis. Okay, so osmosis is the diffusion of water, right? Diffusion of water. So let's take a look at this situation, okay? And if you don't understand, please ask, because I guarantee you will see something like this on the quiz or on the test. Okay, so there's a U-shaped tube here, a U-tube if you would, and there's a membrane at the bottom. Okay, so this membrane only allows water to move through. Okay, I'm gonna have some water here. I'm gonna have some water here. And then I'm gonna put some solute here, okay? Solute is the stuff that is dissolved in the solution. Yeah, so there's some solute. Solute, okay? Now, very important here, please pay attention. The solutes are too big to cross this membrane, okay? So only water will be able to move through, okay? And of course, this whole thing is dissolved in water. I'm just gonna, I don't know why I'm coloring it, but it will remind you that this is an aqueous solution. It's in water, okay? So I'm gonna leave this alone for some time, okay? An hour, 2 hours. And then my question is, what will happen to the water level? Okay, initially the water level is the same, right? So after 2-3 hours, what's going to happen? Okay, is it A, no change? Okay, is it B, the left goes up? Right, the water level is going to go up on the left, and then the right is going to go down. Right goes down. Is it C, the other way around? Right goes up, left goes down. Okay, or is it D, both go down? Okay, I will give you a poll. Just one correct answer this time. Go ahead. Let me know what you think. Okay, remember, there will be a question like this, either on your quiz or on your test, depending on the generator, but make sure you know how to answer it. Okay, a couple more seconds for people who want to participate. Okay, so it looks like we are, again, kind of split between B and C, or half-half, and then we have some people who chose A and D. All right, what's going on here? First of all, the water is the only thing that can move, okay? So the red balls will not move, okay? If the red balls can move, they will move to the right, right? Okay, because it goes from high concentration to low concentration, all right? So what will happen here? Well, nature always try to balance things out. Okay, so as it stands right now, the left side is more concentrated and then the right side is less concentrated. Okay, so how do we balance it out? How do we make it similar? Okay, so one way to do it is to dilute the left side, right? Because it is more concentrated at the moment. And how do you dilute it? Well, if water is the only thing that can move, then water will move to the left, right? And then it will cause the water level to go up. And then similarly, the water level is going to drop on the right side. Okay, so by moving the water to the left side, now you have more volume, same amount of solute, then the left side will be diluted, and then the right side will be more concentrated. Okay, think about like Kool-Aid or something. Okay, like you add the packet and then you add the water and then you taste it. If it's too sweet, you can always add more water to dilute it, right? And that's exactly what's happening here. Right? Initially, it was too concentrated. Okay, so some of the water will move over to dilute it. Okay, and if you remove some water on the right side, it will cause it to become more concentrated. Okay, think about cooking. Okay, you want to keep on simmering your sauce until it thickens, right? Until it becomes this rich, savory, concentrated sauce, right? So by taking away some of the water, okay, you are able to concentrate more. So the correct answer for this is the left goes up and the right comes down. Is there any question about this concept before we move on? No, everybody it's okay. Great. Let's just do. You know what? Let's take a break. It has been an hour. We'll take a quick 5-minute break, and we will come back in a bit. Okay. And if you have questions, please leave it in the chat. I'll see you in 9:38. Okay, let's keep going. What is going on the next page? All right, describe what happens to the cell in each of the following solutions. All right, so we have 3 different solutions, and we're going to put the cell into it, an animal cell. We're going to put it into the solution, and we're going to see what happens. Okay, so this is explained in quite a lot of detail in the lectures recording. So I'm just going to give you like the condensed version. All right, so first one is isotonic. What does isotonic mean? Okay, so I'm going to give you a definition of isotonic. I'll do it in blue, I guess. So isotonic solution means the concentration of solute, okay, inside the cell is equal to the concentration of solute outside. Okay, square bracket means concentration. You'll learn that in chemistry later. So if the outside solution is same as the inside solution, then we call it an isotonic solution. In an isotonic solution, the rate at which water enters the cell, and we'll use like a red arrow for that. Okay, so the rate at which water enters the cell is going to equal to the rate at which water exits the cell. Very important point to remember. It doesn't mean there is no movement of water. It just means that there is no net movement of water. Okay, what does it mean? Okay, so like think about money, right? Okay, if you spend as much as you earn, right? You're still making money and you're still spending money. It doesn't mean you're not moving money at all. It just means that you're spending as much as you're earning. So there's no net change in your savings. Okay, if you spend more than you earn, right, then you have a net spending. If you spend less than you earn, then you have net saving, right? So this is exactly what it means. Okay? Do you gain more water than you lose, or is it the same? Okay? So in this case, if it's in an isotonic solution, the rate of water coming in is going to equal to the rate of water moving out. Okay? Again, it doesn't mean the water is not moving. It just means they're coming in as fast as they are leaving. Okay, you're spending money as fast as you're making them, for example. Okay, so this results in no net water movement. Okay, cell is not gaining water, cell is not losing water. Okay, so what happens in the end? No change in cell shape. Okay. For an animal cell, they are happy to be in isotonic solution. Okay, animal cells, happy. Plant cells, so-so. Okay, they're not unhappy, but they're not like happy either. Okay, plants always want to have more water. This is like if you forgot to water your plants for like, you know, a week or something. Okay, depends what kind of plants, I guess. If you have one of those succulent plants and you only water them like once a month, right? This is like if you forget to water your regular plants, like maybe 2-3 days, right? So it's still okay, but not happiest. All right. So that's isotonic. Next one is hypotonic. What does hypotonic mean? Hypotonic means the solute concentration inside is greater than, okay, this symbol means greater than the solute concentration outside. Okay. And remember, water is always trying to balance out the concentration. So in order to dilute the inside, you will have a lot of water coming in. A lot of water coming in. Okay. More water coming in than leaving. Okay. So rate in is greater than rate out. Okay, so you end up having more water in the cell, right? So net movement of water is net water movement equals to into the cell. Okay. If there's too much water coming in, what happens is, okay, the animal cell, okay, will burst. Another word for burst is it will lyse. Okay, so not good, very sad. And then for plant cell, okay, plant cells, they actually love this. They will be happy. Remember, the plant cell has cell wall. So the cell wall prevents bursting. Okay. So like, think of like my cup here as a cell wall. I can put a balloon inside and then I inflate the balloon. After I inflate the balloon, I can actually just hold on to the balloon and then, move my cup upside down and support it, right? Okay. Because the balloon is creating so much pressure, pushing against the mug, that it could actually support the entire mug. And that's what the plant cell does. Okay? The cell wall is going to create this rigid structure around the cell membrane. And when you have a lot of water in the cell membrane, it's like blowing the air into the balloon, right? It's going to push against the cell wall and it helps the plant stand up. Okay. I don't know if everybody can relate to this example, but like if you buy celery sticks or something, right? You leave it in the fridge for a while, you take them out, you see all the leaves are kind of withered, right? They're kind of like, shriveled up and then they're sad looking. But if you cut the stalk and you put it in the water, right, in a couple of hours, they will open up and they will stand up again, right? And that's because of this phenomena that I'm talking about. All that water is going to then go into the cell, push on the cell membrane, allow the plant to stand up. Okay. So that is a hypotonic. Again, not good for animal cells, right? In the lecture, we talked about did I talk about that person who drinks too much water in the contest? I think I did. Okay, so drinking like huge amount of water actually dilutes your blood substantially, and that creates a hypotonic solution, and it could be dangerous. Okay, there's only certain medical situation where you would infuse a hypotonic solution. If someone is having like a hyper kalemia, too much potassium in the blood or something, then you will flush the system with hypotonic solution. Okay, but generally you would not want that. Okay, so one way to remember this, as I'm sure I talked about this in the lecture, Hypotonic with a big O. Okay, this is when the cell swells up and potentially it is going to go kaboom. Okay. The opposite of hyperpotonic is hypertonic. Okay, hypertonic is when the concentration of solute inside is less than the solute concentration on the outside. Okay, so in this case, the water that are coming in, okay, the rate at which water comes in is less than the rate at which water is leaving. All the water is trying to go outside and balance out the salt concentration. Okay. One way to help you remember this is think about what happens if you take a spoon of salt and you put it in your mouth, right? Okay. You put a spoonful of salt in your mouth. Don't swallow it. You put it there. Like after 30 seconds or something, your saliva gets sucked out, right? That's exactly what's happening here. If there's a high solid concentration surrounding the cell, all the moisture from the cell will get sucked out. Okay? So rate in is less than rate out. So you have a net water movement out of the cell. Okay, so animal cell, okay, they will shrink. Okay. It's called plasmolysis. I don't know if I use this word. I don't remember, but that's what it's called, plasmolysis. Plant cell, okay, also shrink. Okay, not happy. Okay, leaves are going to be withered. Okay, animal cells, not really happy either, but like you know, not gonna die. Okay, they're just gonna shrink. Okay? And one example I used in class was the hypothalamus, right? Okay. Did I talk about this? I'm so sorry. I don't remember if I did. So hypothalamus is a region in the brain that controls all the autonomic functions. Okay, regulate body temperature, regulate your water salt balance in the body, and what happens is there are these cells, okay, in the hypothalamus, okay, and of course with any cells they are next to blood vessels, right? So if you are dehydrated, okay, that means you will have very, very salty blood, so to speak. Okay? Your blood concentration will go up as a result of dehydration, and that will cause these cells to lose water, okay, to the environment. So that shrinkage of the hypothalamic cell will trigger an electrical signal in the brain that causes you to feel thirsty. Okay? And then you drink water. If you drink enough, it will dilute out your blood and it will return it to isotonic once again. Okay, so there you have it, the three different type of environment. Does anybody have any question? Okay, let's do this one. Check all correct answers. So let's see if you can do it without looking at your notes. All right, it has been a minute. Just gonna give you guys a couple more seconds. So of course, on the test, right, it would be beneficial to remember this chart, right, or to have a quick reference sheet with this chart on it, and you can just compare the answers to that sheet. So this is hypertonic. That's the one that the cell will shrink up and the water is leaving. So net movement of water would be out of the cell. That's a correct choice. The cell will shrink because the water is leaving. And the reason the water is leaving is because the solid concentration is higher outside compared to the inside. So the correct choices are A, C, and E. I also received a few emails from students who asked me how come their mark is like, 0.4 or 0.35, some like odd numbers instead of like, whole number or 0.5. That's because of the way that these multi-select questions are calculated. It's actually very complicated, the whole thing. If you want to know, you can email me. I'm not gonna explain it here. Okay, let's see what other things we can do. This one, maybe not yet. Okay, how about this one? Okay, this is another good one. This is just single choice. Go ahead. Okay, I'm gonna end the poll. The correct answer would be small hydrophobic molecules. Right, so you got to think about the factors that affect the array of diffusion, right? So the small, sorry, not necessarily the rate of diffusion, but like think about the property of the cell membrane. Okay, if it's small, it definitely would have an advantage. Okay, so between large and small, the small will always be easiest. Okay, think about squeezing through an obstacle course. The smaller you are, the easier you'll be able to fit through, right? Okay, and think about your cell membrane. Right? The cell membrane is primarily hydrophobic in the center. So anything that is polar charge, hydrophilic, they will have a hard time crossing. Okay, so hydrophobic will be easiest. So small hydrophobic. Okay. Hydrophilic will not be able to cross at all. Okay, I would just want to emphasize that it will not be able to cross on their own. They need to rely on transport proteins. This is application question. Let me see if we can do that now. Yeah, let's do this now. Okay, this one is gonna take some time to read. Okay, I will let you think about that. In case you don't know, you should know because we talked about it in lecture 2. But more H plus equals to a higher, lower pH. Okay, and then less H plus equals to higher pH. In case you forgot about this. Okay, it has been 2 minutes. I know some of you are still reading, but let's give it another half a minute or so, please. Okay. Now, I wouldn't say that this is a hard question. It's just very wordy, and you have to like read it carefully to know what's going on. So we have the student, we have the bag, and it is a highly concentrated salt water. Okay, so that means it has a lot of salt in it. Salt like that. Okay, and then he's gonna put it in a beaker of just water, so no salt, no dots at all. Okay, and the bag is only permeable to water. Right. So what will happen? Well, this is exactly like the hypotonic situation, right? This is a hypotonic solution, okay? And what that means is water is going to go in, okay? And over time, what's going to happen is the bag is going to swell up with water. It's going to increase in volume. So that is the correct choice. All right. Any follow-up question? Okay, thank you for the thumbs up. Okay, do we have another one? Oh yes, this one. Okay. This one is very important for your lab because there's a lab question related to this, and you know, for anyone who did not come to the tutorial or not watch the recording, they will for sure get it wrong. Okay, but you guys came, so you will know the correct answer, hopefully. Okay, everybody should answer, okay? Some of you, unless you're like away from your keyboard or something, or you're actually not there. If you're there, please choose something. Okay, there is a choice for everyone, even if you're not sure, you can choose I'm not sure with the pondering emoji icon. Okay, a couple more seconds. Come on. You can do it. Waiting for 7 more people. 6 more. Okay. Anyone else? 4 more. Where are you? The 4 of you. Are you guys away? Are you there? Choose something. All right. It's pretty good. And the winner is the 2 people who chose C. That is the correct answer. Very good. Okay. So the 2 of you, whoever you are, who chose C, you want to explain to us why you chose C?
I can explain if that's all right.
Of course, please go ahead.
I know that when I kind of applied the same thought that happens with like plant cells or produce when you freeze it to what would happen with animal cells. And when you freeze something that contains water or water in general, water expands. When it's in a cell, it expands beyond the contains of the cell and actually damages the cell wall. So it's essentially already kind of burst open.
Yes, that's excellent. That is exactly the reason. Okay, I couldn't have explained it better than you. Okay, so that is the reason. Right? You ever try to freeze a water bottle or something? Okay, like you fill it up to the brim and then you close the cap, you put it in the fridge, And then the next day you realize the bottle might have burst, right? And even if it didn't burst, okay, even if you didn't fill it up to the brim, okay, and just freeze it, what happens is you see the water bottle swells up, right? Okay, and that is exactly what happens when you freeze anything that has water in it. The water expands, okay, and in this particular case, the ice crystal along with the expanding ice essentially is going to irreversibly damage the cell membrane. Okay, so after you thaw it, you put it in the solution. It doesn't matter what you put it in anymore. Okay, because the cell is like full of holes. Okay, so things just gonna go in, it's just gonna go out, you know, on its own. Things are gonna leak out as well. Okay, so for that reason, and you must remember this when you do the lab. Okay, for this reason, after you freeze something and thaw it, the cell membrane is completely destroyed. Things will just leak out, things will just go in, and then there will be no change in the size. It doesn't matter when we're talking about like a animal cell or plant cell. This is true. The cell wall is not going to protect the plants from the ice crystal. Okay, does that make any sense? If it doesn't, check back the recording later on and make sure you have a good understanding of it. Now you must have a question, right? You must be thinking, if that is the case, how did all those plants like survive the winter? I mean, like, for goodness sake, it was minus 33 degrees like last week or like 2 weeks ago. How is it that the cell of the tree not like all burst and like all died or whatever, right? Okay. Well, it turns out a lot of these, you know, trees and animals in some cases, they produce these natural antifreeze sometimes. Okay, and these natural antifreeze are going to be able to prevent ice crystal from forming, and therefore they will not damage the cells. But if you're talking about like a typical plant that grows in like a normally not an extremely cold environment, they will not have these antifreeze, and if you freeze them, they will die. Okay, they will burst. I mean, okay. Try freezing your spinach. Okay, you take it out and soak it in water, all the green is gonna leak out. Okay, for that same reason. All right, so for all of you who came here today, I expect you to get full marks on a related question in the lab. Okay, excellent. Let's go back to this number 6 right here. Compare active and passive transport. So movement of molecules for active transport, it is going to go from high concentration to low concentration. Okay, high concentration to low concentration. Just like going downhill, it happens automatically, it happens spontaneously, no effort is required. Okay, so this goes down the concentration gradient. Does it require ATP? Nope. Okay, does not require. Oh, sorry. I am going backwards here. Sorry, my mind was wandering away. It should be the other way around. Okay, sorry. Let's start again. Active transport. Active transport is actually movement from low concentration to high concentration. The other way around. Okay, sorry about that. So this is against concentration gradient. Okay, it's like pushing a ball uphill. It requires energy. It requires ATP from the cell. So yes, requires ATP from the cell. Okay. Example of this, we have things like bulk transport. Okay. Things like exocytosis, right? Endocytosis. We talk about that. Another example would be like a protein pump. Protein pump. Okay. So think of like a bacteria that has a special protein pump that allows it to pump out antibiotics. And so you cannot kill it with the antibiotics because the pump will keep on pushing them out. Okay, that would be an example of bulk transport. That would be an example of active transport, okay, using ATP to remove something from the cell. Passive transport, on the other hand, this is going from high concentration to low concentration. Okay, it is down the concentration gradient. Okay, does not require energy from cell. Example would be osmosis, right? Simple diffusion, right? Facilitated diffusion. Okay, for those of you who need a refresher, facilitated diffusion is when the substance that is trying to cross is too bulky. Okay, you cannot go through the phospholipid bilayer on its own, or maybe it's charged, then it's going to require the help of a membrane protein. So that's called facilitated diffusion. It does not require energy. The protein just provides a passageway for them to go through. Okay. Next one. Choose the more concentrated solution in each of the following. So we have 80% water, 20% starch versus 90% water, 10% starch. When we check for concentration, we can look at the solute. In this case, it's the starch. So the more starch you have, the more concentrated it will be. Okay. B, we have 5 grams of sodium chloride in 50 grams of water versus 5 grams of sodium chloride in 25 grams of water. Okay, so to some people, this might be obvious. If it's not obvious, maybe the math will help. Okay, so let's see. 5 grams of NaCl in 50 grams of water, right? Okay, so if I reduce this, for example, if I divide the bottom by 2, I divide 50 by 2, I get 25 grams, right? Okay. But if I divide the bottom by 2, I must divide the top by the same number, right? So 5 divided by 2 is 2.5 grams of NaCl. Right? So the first solution is equivalent to 2.5 grams in 25 grams of water. Compared to 5 grams of salt in 25 grams of water, this one has more salt. So that is more concentrated. Okay, you want to try to get the amount of water the same by either multiplying or dividing, and then you compare the solute. Okay. Last one, 85% solvent and 15% solute versus 75 and 25. Again, we are focusing on the solute, and this one has more solute, 25%, so that's more concentrated. Okay. Three types of bulk transport. We have, phagocytosis. Okay, that's like cell eating. Right? When you have a white cell here, there's a bacterium. It eats it up, right? Like Pac-Man. That's phagocytosis, cell eating. Okay, so your immune system does that a lot. Another one is pinocytosis. That's cell drinking. Instead of taking in like a solid chunk of stuff, you're bringing in just a fluid. And then you have things like exocytosis. Okay. There's also another kind called receptor mediated. It's in the notes. Endocytosis. So this one, it adds more specificity to it. Specificity. Sorry, specificity to what can be brought into the cell. Okay, it must first match the receptor, then it's going to trigger the endocytosis. All right. Okay, any question about these particular ones, the long answers? Do you need me to go through it? Any requests? We are a bit tight on time, so if you don't have any requests, maybe I will just post the answer to these ones for you to look at. Let's just quickly look at the application questions, and then I'm going to talk about the lab. All right. First one, I think I already talked about this in the lecture notes. Okay, but basically, this is a real thing that happened. Someone in a radio contest to win like some prize. She drank a lot of water without going to the bathroom. Okay, she won the contest, but later was found dead in her apartment because of water intoxication. So how does drinking a large amount of water in a short period of time results in water intoxication? So a couple of things here. If you drink a lot of water, okay, you are diluting your blood, diluting the blood. Okay. So that could lead to RBC, your red cells, right? And even your white cells bursting. Okay, and of course, the red cells are important for transporting oxygen, right? So if they start dying, then you won't be able to circulate enough oxygen. But that's not the main reason. The main reason you die from having too much water is you are diluting electrolytes. Okay, electrolytes are things like sodium, calcium, right? Chloride. All these things are actually very important for normal functions. Your nerve cannot fire, cannot communicate with each other if the sodium level is not correct. Your muscle will not contract if you don't have enough calcium in them. So if your heart muscle cannot contract, like you die, right? So I was going to ask, how much water do you need to drink to get to that point? You know, it's quite a lot, right? You will feel sick, right? Before it leads to a point where you're gonna die. Okay, we're talking about like maybe liters, more than a liter of water at a very short amount of time. Okay, normal, like your body is amazing, right? Like it will tell you something is wrong, right? So if it feels wrong and then you keep on doing it, then that pushes the body outside of the safe zone, right? Beyond the point of return, so to speak. Okay, so, but this was like a contest, right? So the person probably was not feeling well with all these water coming in already, but then she keep on pushing it because she wanted to presumably win the contest, right? So there will be warning signs before you get to the dangerous zone. So not to worry about it. Just drinking your. People say drink 8 cups of water a day, right? That's fine. That's not going to cause you to have water intoxication. Remember, this has to be done in a very short period of time, right? The other one is single cellular organism that lives in fresh water are constantly exposed to hypotonic solution. Speculate what kind of transport mechanism could be used to prevent lysis. Remember I talked about this little guy last time, right? This paramecium. Okay, paramecium. It's a live in a fresh pond water, and what happens is water will keep on coming in because it's hypotonic, right? So it has a special pump. Okay, it's called a contractile vacuole, and it will constantly be pumping water out of the cell. Think of this as a leaking boat. Okay, so you have to keep on scooping the water and then pouring it out so the boat does not sink. Essentially, that's what this pump does. Okay, it constantly uses ATP to remove the excess water coming in and prevent it from bursting the cell. Number 3, salting has long been used as a method of preserving food. Coating meats with layer of salt will dehydrate the meat as well as any microorganism. Explain how this process works. So it's very obvious, right? I hope it's very obvious. By having a lot of salt, you create a hypertonic environment, right? Okay, so that sucks all the moisture out of the cell, right, out of the microorganisms, and essentially kills them. And without microorganism, the food is not gonna it's not gonna go spoil. Okay, so that is essentially how you make curated meat. All right. Yeah, so that is that. If there is no additional question, I would like to quickly talk about the lab. I know we are a little bit tight on time. If you do have to go, please feel free to do that. And you can watch the recordings afterwards. Okay, so here is what's going on with the lab next week. Okay, you should have access to it right now. And if you don't, I will make sure it is available after class. All right, so I'll post an announcement that gives you a quick link to the lab. But this is how you get to it. Click on content. You are going to scroll all the way down to virtual labs. Okay, so there is a video and then there is the actual lab that you have to fill in. Okay, do not try to do the lab without watching the video. Okay, a lot of the answers to the questions in the lab are actually in the video. Right, so if you would just take the time and watch it, it's only like 10 minutes or something, you would already guarantee to get like a B+ or something in the lab. Okay, what happened in the past is a lot of people just wing it. Okay, they don't do the lab until like it's Thursday, and then they just try to answer the question by Googling the answer. It will not work. All right, the lab is going to be due next Friday at midnight. Okay, so you have like a whole week. My suggestion is just get it out of the way. Okay, so study for the quiz. Okay, and then after Tuesday you do the quiz, just do the lab and then hand it in. All right, yeah, the deadline is next Friday, 11:59 p.m. or something. Okay, but it's very easy. The lab should take you no more than an hour. You sit down, you watch the recording, and then you do it. Okay, so how do you do it? You click on the Beetroot Lab. You will have to download it. So there's a download button. Okay, so you go ahead and download it. It's going to be on your desktop or somewhere wherever you save it. Okay, I'm gonna save it on my desktop over here, and then you can open it. Hey, if you use Apple computer, I think you can still open this. If you have trouble opening it, let me know. I'll send you like a link to a Google Doc or something like that. Okay, so all you have to do is again, it's related to cell membrane structure as well as scientific methods. You can watch this video. It's the same link on the website, and then you complete all the questions here. Okay, so the first question: what is the purpose of this experiment? Okay, so you type the answer in the box. Okay, so like this. Okay, the purpose of this experiment is to investigate the effects of temperature on membrane permeability. Okay, so there, I've given you the answer for the first question. So everybody should get 2 marks. Okay, that's it. Okay, you don't have to type me a paragraph. Okay, keep it short, keep it concise, keep it clear and correct. Okay, people keep on rambling on and on and on in their answers, and that makes it very difficult to mark. Okay, just try to answer it in 1 or 2 sentences at most. Okay, so what is the hypothesis? You write a hypothesis. Remember, a hypothesis is a statement. Do not give me a question for hypothesis. Okay, many people in the past wrote, will temperature affect the membrane permeability? That's not a hypothesis. You're supposed to tell me that whether it will or it will not. Okay, and then independent variable, dependent variable. Most of these are pretty self-explanatory. Okay, safety precautions. Okay, don't give me things that are not related to the lab. Okay, for example, people say what do they say? Like they like, you know, watch out for acid burns. We're not using acid in this lab. Okay, so just use things that are relevant to this lab. Okay, and also don't be repetitive. Okay, if you say wear PPE and then the next point you say wear goggles, well, goggles are PPE, right? So don't repeat yourself. Make sure that these are distinct things, and try to think about it when you're actually doing this lab. What kind of precaution are you going to be taking? Okay, this question is another one that people get confused. Again, limit your answer to 3 or 4 sentences, right? So don't go on and on, right? Just answer the question and be done with it. So here there are 2 questions. Number 1, which test tube is considered as the control of the experiment? So you would tell me by saying the control of this experiment is, there are only 4 test tubes here. Is it the 20 degree? Is it the 40, the 60, or 80? Make your choice. And then explain to me why we need to have a control in an experiment. Okay, if you don't remember what a control is, revisit the first lecture. Okay, so you answer both of these parts to get 2 marks. Okay, and again, these are self-explanatory. Okay, here is another one. And again, right, if you don't catch everything I'm saying right now, because I'm going a little bit too fast, watch the last part of the recording again and make sure you understand it. Okay, describe the correlation you observe between temperature and color intensity. Okay, many people seem to not understand what I'm asking here. I am simply asking what happens to the color as the temperature goes up. Okay, so you would answer by saying, as the temperature increases, the color intensity, and then fill in the blanks. Does it get more intense? Does it get less intense? Is there no change? That's all I'm asking. Okay. Now, is that qualitative analysis or like, is it quantitative, right? What is the difference? Again, this is covered in the first lecture. If you need a refresher on what the difference between qualitative and quantitative means, just revisit the lecture. Okay, don't Google it. Okay, you Google it, you're gonna end up with some answers that is not necessarily applicable to what we are talking about. Okay, just go to the lecture notes and look at it that way. Describe the relationship between absorbance and spectrophotometer. Again, the person in the video will explain what this means. I think she used a slightly different word. She said a calorimeter. Okay. It's similar stuff. Okay, they both measure the color. They give you a number instead. Okay, so for example, like to you and I, 60 and 80 might look the same, right? They look similar red. Okay, but if you measure it with the calorimeter or the spectrophotometer, it will give you a specific reading, a number. Okay, so like this might be, I don't know, making this up, like 450, and then this would be like 380. So there is a difference. Okay, because we're giving a number to it, we call that quantitative, right? As opposed to just using our senses. And one more. This one is usually where people lose a lot of marks as well. Okay, this asks you to plot the data. Okay, so in your computer class, I believe they already taught you how to do like a graph in Excel. Okay, if you don't have Excel, you can also use like Google Sheets. By the way, I don't know if you know, as students of the Centennial College, you are entitled for a free copy of the Microsoft Office. Okay, you can go on to your My Centennial site and you can download a free copy of it. Okay, so if you want to do that, you could. If you don't, just use Google Sheets. Do not draw me a graph and send it to me. Okay, that is not accurate and it does not meet the standard that I want. Okay, so do a graph on the computer and make sure you label the x-axis and the y-axis. Okay, so many people give me a graph, but they did not label it. Okay, if you need help of how to graph, you can just watch this video. It basically guides you step by step on how to do it. And finally, make sure you use the right type of graph to display this data. Okay, I will tell you a pie graph, the circle graph, is not the right type. Okay, many people use circle graphs in the past. I'm not sure why, but that would not be the correct type. Okay, so think about what kind of graph to use. In fact, if you watch the video shows you what kind of graph you should be using. Okay, and then there is some data analysis. You see here is the frozen beetroot question, right? That many people did not get right, but you came here, you did the application question, you should be all right. Lastly, if you are confused about any of the questions, just send me an email. I'll clarify for you. Okay, I want everybody to do well on this lab. Okay, it's 10%. It will boost your grade tremendously if you didn't do so well on the quiz. And if you did well on the quiz, this will maybe boost you from an A to an A plus. Okay, so this is what you have to do next week. And once you type up all the answer, you save it. Okay, I'm not going to save it, and you will have this copy on the desktop, and then you will submit it. Okay, I just want to know, did your other teacher tell you how to submit things on eCentennial? Do I have to go through that now? Do I have to go through how to submit assignments on eCentennial? Okay, great. People seems like already know how. I will provide a link for you to do that as well. If you cannot submit it for some reason, just let me know. Okay, but don't email me your assignment. I cannot grade it from my email. Okay, I need to grade it within the system so the marks can be uploaded. Other than that, good luck with your quiz. And again, no class next week. I will see you in 2 weeks. Have a wonderful week, and stay safe in the storm. Okay, bye for now.